This report presents an executive scientific overview of top intellectual property positions in mRNA-encoded bispecific antibodies. It's part of a larger series that evaluates the state of gene therapy, cell therapy, and antibody innovation. I've broken down this analysis into four key pillars: scientific opportunity, clinical proof of concept and real-world evidence, economic potential, and market access. To read this report in its entirety, please refer to our website. Please click the link provided to access it there. And while you're on our site, make sure to subscribe to receive future reports like this one. 1. Scientific and IP Landscape Overview. The integration of mRNA technologies with bispecific antibodies is an emerging therapeutic paradigm that promises to solve key limitations in biologics manufacturing, targeting flexibility and tumor heterogeneity. mRNA enables in vivo expression of complex bispecific constructs, bypassing many of the bioprocessing bottlenecks associated with recombinant protein engineering. Top 5 relevant IP assets as of 2024. 1. Moderna Inc. US 11739-185b2. This patent describes a delivery platform encoding bispecific T-cell engagers targeting 𝗕𝗓𝗢𝗏 and CD3, delivered via lipid nanoparticles. Early animal models demonstrated tumor shrinkage with reduced systemic toxicity. 2. Biotech 𝗕𝗜𝗢𝗏𝗘𝗦. This patent covers mRNA constructs encoding bispecific antibodies for immune checkpoint inhibition, e.g., PD-1-XL8G3, with tailored UTR and codon optimization to enhance translational efficiency. 3. Pfizer Inc. W020-318-847A1. This patent claims synthetic mRNA compositions encoding bispecific antibodies for hematologic malignancies, designed for or intratumoral delivery using PEG lipid-stabilized LNPs. 4. Arcturus Therapeutics, US 20230311 for 32A1. This patent involves circular RNA circRNA encoding bispecifics, prolonging expression in vivo. The patent includes formulation for liver and spleen targeting, with sustained protein production. 5. CureVac D1020-11199 for 1A1. This patent focuses on mRNA encoding by its bispecific T-cell engagers targeting prostate-specific membrane antigen PSMA and CD3, with LNP formulations adaptable to multiple tumor types. Emerging trends in this space include tailored UTR and codon optimization for enhanced translational efficiency, synthetic mRNA backbone modifications for stability and immunosilencing, formulations for targeted delivery to tumor sites and immune cells, modular designs enabling rapid iteration and personalized medicine approaches. 2. Clinical proof of concept and real-world evidence. While clinical stage assets are still early, preclinical models have demonstrated robust T-cell engagement and tumor regression in xenograft models using 𝗕𝗓𝗢𝗏 and EGFR-CD3 targets. mRNA-encoded bispecifics show superior pharmacokinetics compared to recombinant BSABs due to intracellular expression and absence of Fc-mediated clearance. Additionally, modular adaptation to tumor microenvironments has shown promise in overcoming cold tumors through checkpoint modulation, e.g., PD-L1-XIL to bispecifics. Bite-style constructs delivered via mRNA also reduce systemic cytokine release syndrome observed with IV protein-based T-cell engagers. Real-world evidence (RWE) from mRNA vaccines. COVID-19 has validated LNP-based systemic delivery in millions of patients, supporting safety and feasibility. Furthermore, regulatory familiarity with mRNA platforms will expedite approval processes for BSAB constructs. 3. Economic potential. The mRNA integration holds significant economic promise by addressing key pain points in BSAB manufacturing, development, and commercialization. Cost advantage. Traditional BSAB biologics require complex cell line development, co-expression tuning, and multi-step purification. mRNA-based production cuts production time from years to weeks or months. It also enables on-site or regional manufacture via modular mRNA synthesis and formulation facilities. Importantly, this approach reduces production cost per dose by 40 to 60% compared to recombinant protein-based bispecifics. Speed to market mRNA-encoded BSABs can progress from target selection to phase 1 trials in 6 to 12 months, compared to 2 to 4 years for traditional bispecifics. This offers a critical first-mover advantage in underaddressed indications. Market size. The global bispecific antibody market is projected to reach $1 billion plus by 2035. mRNA integration could expand, and this by $6 with specific value in solid tumors with immune escape mechanisms, relapsed, refractory hematologic cancers, immune modulation in autoimmune and infectious diseases. Strategic deals and licensing already there has been $100 million, $500 million upfront paid for licensing of mRNA-encoded constructs and delivery systems with additional milestone and royalty payments. This approach aligns with the goals of biotech pharma partnerships seeking scalable, high-value oncology assets. 4. Market access and positioning. mRNA-encoded BSABs are well-positioned to meet payer requirements by consolidating 2 or more monoclonal therapies into a single product, lowering cost and simplifying administration. Health economic modeling suggests a 10 to 20% reduction in annual therapy cost, particularly in oncology and immunology, making the approach more attractive to payers. These therapies are also well-positioned for FDA Fast Track and Breakthrough Therapy designations, EMA Prime status for high-need conditions, orphan drug incentives in rare or resistant tumor subtypes. This may reduce time to market by up to 50%, enhancing return on investment and payer acceptance. Finally, mRNA sequences can be personalized rapidly, allowing use in neoantigen-specific constructs. The modularity of mRNA design supports multiple iterations with minimal platform revalidation, unlike protein-based methods. 5. Competitive landscape. The fusion of mRNA and bispecific antibodies is emerging as a platform battleground, with major players positioning themselves for dominance. Biotechne leverages protein expertise to support bispecific discovery. It likely will commercialize custom BSAB libraries and develop GMP-grade mRNA templates, serving as a CDMO or licensing partner for biotech innovators developing mRNA-encoded BSABs. Pfizer has deep mRNA infrastructure via the Biotech collaboration. Internal programs already focus on T-cell engagers for hematological malignancies. We expect expansion into solid tumors and checkpoint modulating bispecifics using mRNA vectors. 6. SWOT analysis strengths. Rapid design, prototyping and manufacturing, lower cost and higher scalability than recombinant biologics, ability to express complex or unstable bispecific formats in vivo. Weaknesses. mRNA stability and delivery remain limiting in certain tissues. Uncertainty in long-term immunogenicity of repeated mRNA bispecific exposure, limited clinical data compared to traditional formats. Opportunities. Personalized oncology expansion, autoimmune modulation, mRNA design and smart LNP integration with AI, attractive for orphan and niche indications with rapid turnaround needs. Threats. Delivery technology, IP wars, LNP patents, etc. Competition from next-gen protein formats, trispecifics, CAR-NK, market skepticism until phase 1 half efficacy and safety data mature. 7. Strategic forecast. If these novel mRNA-encoded bispecific products are successfully developed, we forecast the following. Economic gains. High-margin products with potentially multi-billion-dollar market penetration, especially if first-in-class or best-in-class in key indications. Market access, rapid clinical development, payer-aligned value propositions, and streamlined manufacturing access will provide commercial advantages in time, cost, and patient reach. Competitive winning. Developers with integrated capabilities target discovery, mRNA engineering, delivery science, GMP mRNA production will dominate licensing and commercialization. We believe this modality represents a strategic imperative for both biotech startups and large pharmaceutical players, particularly in oncology and immunology, where dual target or immune synapse modulation is critical. Thank you for watching. To view this report in its entirety, please visit our website. While you're on our site, don't forget to subscribe to be notified of our latest content.